Namaskar. I welcome to our channel Wisdom Coaching Center. As a part of lecture or as a part of series of lecture, second part of that first video, what I told you that to be continued. I explain you what is diploblastic acylomate, diploblastic acylomate, triploblastic acylomate, triploblastic pseudocylomate and triploblastic coelomate. Now the question comes then, what is it? For that explanation, separate video that we are going to have it, that is how these three germinal layers are developed. What is common among all organisms? Why they have this explanation? Let us understand. See, every organism tend to reproduce by the process of fertilization. Here, suppose this is the sperm and this is the ovum. This sperm and ovum that they do reproduce, they do fertilize, they do combine and have the fertilization. So sperm and ovum, that syngamy, fusion of these two commit, fertilization leads to produce a zygote. Now this is a zygote. Zygote undergo mitosis and produce two cell stage. Through animal and vegetative form, it get divided into two cells. Now here, this continue to have that diploid condition. Then produce four cell stage. Size is same, but that divide into four cells. Suppose these are the four cells. Size of the zygote is same. Then 8 cell stage. Here it will have 8 cells. Now division can be shown graphically. So these are the 8 cells. This is 2 cell stage, this is 4 cell stage, this is 8 cell stage. Now 16 cell stage. Here again, to understand, the first division was straight, second right angle to one, third periclinical, fourth also periclinical, having 16 cells. And now this stage is called as morula stage. This is a morula stage. And after morula, then come 32 cell stage. By the time when these cells undergo mitosis and produces 32 cell stage by the time itself. Now here, this is called morula stage, having 32 cells in it. Each cell of this stage is called blastomer. Each called blastomer. So these are the cells. Each cell is called blastomer because these stage undergo blastulation. This is a embryo stage with 32 cell stage, undergo rearrangement of these cells. 32 cell stage, complete morula. Now these cells undergo blastulation, that is arrangement of these cells to form the blastula. What is blastula? As that cells are rearranged. Here having this tropoblast, some of the cells they form outermost layer called tropoblast and form inner cell mass. So here some of the cells they get organized to one side or one pole and form inner cell mass or embryo proper. So these are the embryo cells. Embryo cells, inner cell mass. Inner cell mass. Now here this is tropoblast. This is called tropoblast. Now see, next stage is supposed to have the rearrangement of these cells into 3 germinal layer. Here the beginning of evolution, that 2 derm, 3 derm and 2 derm without coelom, 3 derms without coelom, 3 derm with pseudocoelom, 3 derm with coelom. Now here these cells, inner cell mass, they arrange into 3 germinal layer, and this tropoblasts get anchored into the uterus by forming chorionic villi. Now just for one cell I show how chorionic villi are developed like this, and that get implanted into the uterus, but these 3 continue to form 3 germinal layer, and that stage of conversion of these blastula, This stage is known as blastula. This is called blastocele. This is called blastocele. This is inner cell mass. This is trophoblast. Now gastrulation begins. Now here, gastrulation is only the stage where that it decides that whether they are diploblastic coelomate or triploblastic coelomate or triploblastic pseudocoelomate or triploblastic coelomate. Now to have arrangement in porifera and coelenterata. See, I am going to make that 4 part. Porifera, coelentrates, ctenophora, they have diploblastic coelomate. They have diploblastic coelomate. And diagram is already you have seen in last part of the video. Here, second arrangement, that triploblastic coelomate. Triploblastic coelomate. Second arrangement, and that is you have seen in Platyhelminthes. Platyhelminthes. That you have seen triploblastic coelomate. Then Aschelminthes. We have seen or we have studied that triploblastic so here, and fourth arrangement is found annelids onward. Annelids onward, all phylum or classes, phylum, they have triploblastic coelomate, and that diagram we have drawn. So here it becomes very clear to us that these stages, up to this stage, is common in all organisms, right from porifera to mammals. But from this stage, it changed to whether in that evolutionary order that it is whether diploblastic coelomate or triploblastic coelomate or triploblastic pseudocoelomate or triploblastic coelomate. And that is what we had already drawn and understood. This part of the video is going to make you as a conceptual knowledge of how that diploblastic arrangement of the germinal layer into 2 or 3 germinal layer. These diploblastic, they consist of ectoderm and endoderm. Triploblastic, these they consist of 3 layer: ectoderm, mesoderm, endoderm. So mesoderm came into existence as a part of evolution without cavity, with false cavity and with true cavity. Here, as a part of clearance, that mesoderm with triploblastic, no doubt, mesoderm without cavity. Here, mesoderm with, in this, mesoderm with false cavity or fragmented cavity, not false, fragmented cavity. And here mesoderm with true cavity, mesoderm with true cavity, true and complete cavity. Here we clear now that as a part of evolution, how organisms are evolved. It set into their embryo development itself. Now, as you know that they have triploblast, means mesoderm, endoderm and ectoderm what present in porifera, that is directly as a evolution transfer to this one. Therefore, step by step, organism becomes evolved, and therefore, we mammal as a highly evolved organism on the earth. They are developed from that unicellular form what we explained in protozoa, protista. So it is a long history of evolution that is set into this development itself, that we could understood that how that different layer has role to how the different level of development of organization and in organism. So multicellular without tissue level, multicellular with tissue level, multicellular with organ level, and multicellular with poorly developed organ system level, and later multicellular with well developed organ system level. As the part of the second part of video, that I was supposed to explain you how these organisms with this type of embryo are produced from a common structure called zygote. It is common up to this Gastrula. Yes, and this has to be understood as a conceptual part of study. If this is not being explained, this is, although you have mug up that diploblastic coelomate, triploblastic coelomate, triploblastic pseudocoelomate or triploblastic coelomate, but without this knowledge, you are not supposed to going to enjoy the further explanation of this phylum. Therefore, it was my responsibility and duty to make you known for this part of the knowledge, so that now when this word comes, you will have complete knowledge. That is clear figure in front of you. I hope you would have liked this explanation in addition to your syllabus, and I am happy to have introduction of this knowledge to you, so that you will be aware about the future developments of these organisms in a series of evolution. I am happy to be with you with this part other than syllabus. I hope you would have liked it and enjoyed it. Still, if you are in doubt, you can ask in comment section. I appeal you to subscribe it, and by pressing bell icon, you will be notified as and when that new videos will be uploaded. Thank you.